Dear students, in this topic, we will talk about two major groups of excavates. These are Diplomonads and Parabasalids. Just like typical excavates, these two major groups, they are also present in anaerobic environment. Anaerobic environment means that an environment where there is no oxygen. Now, these protists are very specialized that they lack plastids, due to which they cannot synthesize their own food. They also lack mitochondrial DNA. They also don't have electron transport chain present in their mitochondrial DNA, and they also lack different enzymes which are needed for the citric acid cycle inside the mitochondria. Now, here are the picture of some of the specialized diplomonads. These diplomonads, typically they don't have mitochondria, but if they have mitochondria, they are very small and they produce cofactor for enzymes involved in ATP production in the cytosol instead of mitochondria. Now, these diplomonads have two equal-sized nuclei and they have multiple flagella which help them in order to move from one place to the other place. Now, most of these diplomonads, they inhabit the intestines of mammals. So whenever we have grazing mammals, whenever they go to some streams, even the stream which is contaminated with the feces of those mammals, they can contain these protists. Now, Giardia is one of the typical example of these diplomonads. So even if a stream with the very clean water in the mountains, it could be contaminated with the feces of different mammals, and those mammals can have Giardia in their feces. Now, if it is contaminated and if some humans drink that, it could result in severe diarrhea problems. Now, boiling these stream water can kill these microorganisms, these parasites. Now, another parabasalids, when we talk about them, there is a major group which is called dicycristates. They are basically zooflagellates. They are named after a disc-shaped mitochondrial cristae present inside their mitochondria. They include euglenoids, which are unicellular, and they are mostly photosynthetic. They have plastids in their body and they can synthesize their own food. They generally have two flagella. As you can see in the typical euglena examples, we have a very small one small, which is very short one. It is short flagella. And then we have a large flagella, which is quite large and protrudes outside the body of these euglena. Whip-like movement of this flagella helps euglena to move from one place to the other place. The mode of reproduction is mostly asexual, and the simple mitosis is the process by which it reproduces. Some photosynthetic euglenoids, when they are placed in dark for a longer period of time, they can lose the ability to synthesize their own food. So they become heterotrophic, which means that they absorb organic molecules and break them down to make food or to create energy for their survival. Now, some other species of euglenoids, they absorb organic molecules, even they engulf bacteria and other small protists to degrade them for their energy requirements. Now, these all food particles or the bacteria or other protists, they are absorbed in their food vacuole where they are digested and help in the generation of energy for these parabasalids groups.